Hello everyone, my name is Nathan Stevens. I am a senior at Brooklyn High School. Today I will be presenting a poster for the project that I am currently involved in. I got the opportunity to be involved in this project work under my mentors Sharon Padul and Dr. Asela through the Young Exceptional Scholars Program. The topic for my poster presentation is mapping transcription factor interaction in the soybean. The soybean is an important crop for the U.S. It carries rich nutritional and economical value. By studying transcription factors and their interaction with each other helps us understand the gene expression of the proteome in a soybean. The study of their transcription factors is very important because it allows us to generate superior plants in the sense of defensive productivity. We are focusing our project on defense-regulated transcription factors and their interaction during gene regulation. In Figure 1, we can see that the transcription factor binds the binding site and helps the gene to regulate, eventually to produce a different protein. In Figure 2, you can see how two different proteins are interacting with each other to bind to the DNA to express that gene. And in Figure 3, demonstrates how molecular cloning works. This figure shows how we can insert the gene of interest into the vector, which will transport the gene of interest to the competent cell and multiply vectors when cultured. Vectors are DNA plasmids found in bacteria. The goal for this project is to establish a protocol to study the protein-protein interaction between defense-regulated transcription factors and our interest. Specifically, our objectives for this project are to clone genes into GST and halo-tagged vectors and define 8 protein-protein interactions between these transcription factors. We began by extracting 8 open reading frames from our soybean, and then it was cloned into our donor vector using BP cloning method. Now we call this donor vector an entry clone because it has our gene of interest. These entry clones were then transformed into E. coli and were cultured in LB plus kanamycin media. Since this vector is resistant to the kanamycin, it will allow cells to grow in this media. After the incubation of the media, colonies of PCR was done to detect the clone vector and gel electrophoresis. After this step, plasmid extraction was done from the positive colonies. The inserted gene in the entry clone was then transferred to the destination vector using LR cloning. Since the vector is resistant to carboxylin, they were cultured in LB plus carboxylin media to ensure growth of the cell correctly inserted with the destination vector. After colony PCR and purification, we sent these plasmids for sequencing. After getting our results, we compared our sequences with a database using BLAST. This is our entry vector that we used. This is our destination vector that we sequenced with our genes of interest. In Figure 4, we can see the results of our gel electrophoresis. This showed us that the colonies were good for the sequencing. In Figures 5 and 6, we can see the BLAST results that showed us the GST tag and open reading frame were inserted into our vector and were aligned with good score. Looking into these results, we could conclude that the open reading frames were cloned and in the vector, the results were good. In the future, we are aiming to detect transcription factor interaction using the wheat germ extract, followed by the pull-down assay for our final detection. In this step, we will be tagging our protein with both GST and Halo tags. Finally, I want to thank 2020 Young Exceptional Scholar Program for this opportunity and the funding, Arkansas State University and Arkansas Biosciences Institute for the facilities and support. Thank you everyone for listening to my oral poster presentation. I hope you have a nice day.